is a kind of, network, all right, of protein fibers, okay? You have to know there is two types of protein. في fibers اللي هما they are like rope-like structures, okay, filaments. And there is other types of protein which are called globular protein. It's like a ball, it's like soccer ball, all right? في globular وفي filaments, all right? So the cytoskeleton, there are protein which are filaments, okay? And they are interconnected, they form what we call a network, all right? And this network, as you said, is very important for the structural shape of your cell, okay? It's very important for the shape of your cell. And it is also, as you said, it is very important for the anchorage of the organelle on the cytoplasm, because we said the organelle, they don't float in the cytoplasm. They have positions which are determined by these cytoskeleton, okay? And also, we will see some of these, we can say, cytoskeleton, we can say these fibers, they are very important for the motility, for the movement of certain organelle from one side of the cell to another side. And now what we said they are fixed, they have their positions in the cytoplasm, they cannot. But to move, they can use these microtubules or these cytoskeleton fiber to move from one side to another side. So we will see this later on the side. So the things that we have to say as well is that these or this cytoskeleton, it is present in the cytoplasm, it is present in the nucleus, but not in the other organelle. بس في النيوكليوس وفي السيتوبلازم. Okay? So this is very important to know. Okay? All right. So as you said, we have three types of cytoskeleton or protein fiber. We have the microtubules, we have the intermediate filament, and you have the microfilament. So I present them like that, first one, the second, and the third, because they are classified according to their thickness, okay? So the thickest one is the microtubule, then the intermediate filament, and the thinnest one are the microfilament, okay? So they are, we can say, arranged according to their thickness, okay? Is it clear? All right. So now, today, in these lectures, we're going to focus about the microtubule. And next Monday, inshallah, so we're going to talk about intermediate filament and also the actin filament, or we call microfilaments. Okay? Do you have questions so far? Yes?
So the nucleus has a cytoskeleton.
Huh?
The nucleus has a cytoskeleton.
Yes.
But we mentioned that the organelles in the nucleus float around. So what's the function of it? Why is that important?
You will see it now. Okay? All right. So let's start by the microtubules. As we said, it is the thickest one, all right? The thickest, we can say, protein fibers among the others, and which make them, this property make them very strong, and they resist any compressions on the cells, okay? So this, it helps the cell to maintain its shape, okay? So the cell, sometimes it exerts some compressions, okay? Some swallowing of the cell, some shrinking of the cell. So all these, they try to resist to the compressions which is exerted on the cells, okay? So there is two types of microtubule. There is what we call the long-lived and short-lived. Can you tell me what does it mean? What does it sound for you? Short-lived, long-lived? Yes?
Lifetime?
Lifetime, yes. So what is it? Is lifespan? Yeah, we can say this kind of lifespan. Long-lived, it means that these microtubules, they will exist in the cell permanently. Okay? They will exist permanently. All right? The one which are short-lived, so they are, we can say, synthesized, they are produced by the cell to ensure a function. Once this function is finished, they will disappear. Okay? So the long-lived, they will live very long. Okay? They will stay in the cells. They will not move from there. All right? The short-lived, they are there for a mission, for a functions. Once this function it is ensured, they will disappear. Okay? So the one, for example, the long-lived flagella, cilia, etc. So these, they are present permanently. All right? However, for example, the short-lived, one of the example is the spindle fiber. Do you know what is the spindle fiber? Or mitotic fiber?
It's for meiosis, I think. When it's
Yes. The spindle fiber, now when I come to you by saying there is a cytoskeleton in the nucleus, is that during mitosis, the cell create what we call the spindle fiber, where the chromosome, they will be attached. Okay? وبعدين تصير لهم after that you're going to have ascensions. Okay? So you have the prophase, metaphase, anaphase, telophase. So during this mitosis, we're going to have like a spindle fiber. Okay? I'm going to show you the picture here. This one. Okay? And here you have the chromosome in blue. So the chromosome, they will be attached. All right? And then we're going to have after that separations of the chromatid and so on. All right? So this, we can say spindle fiber, it is made of microtubule. Clear? So once this microtubule, they made the spindle fiber, once the cell divided and the division is finished, it will disappear. Okay? So that's why we say it is short-lived microtubule. واضح? All right, so what are the function of microtubule? As we said, it is a component of spindle fiber. The second thing also as structural support. For example, we find it in the cilia, in the flagella, you know, so we can find it there. And also, the third thing, which is very important, is the intracellular motility. What does it mean, motility? Movement. Is the movement inside the cell. All right? You remember I said the organelle, they are stuck, you know, within the cytoskeleton. To allow them to move, they will use the microtubule as a route, as a track, all right? Allow them to move from one side to another side, okay? So this is one of the function of the microtubule, and we're gonna see it. We're gonna watch a video where we're gonna show how this microtubule, they will be used to allow the movement of the organelle inside the cell, okay? Is it clear? Yes?
And I think it transports some molecules.
Sam?
Some molecules.
Yes. Okay? Is it clear here? واضح? All right. So now we go to the things more in deep. So microtubule. As you can see here in the pictures, the microtubule is a kind of cylinder. It is empty inside, so that's why we call it hollow cylinder. Okay? So it is a cylinder shape, and inside it is empty, so that's why we call it a hollow cylinder. All right? So what is the structural unit? From what it is composed, your microtubules? The microtubule to be formed, they need the globular protein, which are called tubulin. So the microtubule, they are composed by tubulin. So the tubulin, then when they will assemble, form these cylinder. Okay? You have to know that we have two types of tubulin which make the microtubule. We have alpha tubulin and beta tubulin. And these two subunits, they will associate together and they form what we call a dimer. Tubulin dimer. You know what does it mean, dimer? Yes. So there you have to know monomer, dimer, trimer, tetramer. So this is the tri, the mono, the di. It is related to the number. Mono is 1, di is 2, tri is 3, tetra is 4, okay? And the mer is related to the subunit. How many subunit? Okay? So if I have monomer, it means 1 subunit. So if I have a dimer, it means 2 subunits. Okay? So here I have 2 tubulin, alpha and beta, so I will have a dimer. Okay? Clear? Okay. So my microtubule, as you can see here, it is composed by 2 globular protein, alpha tubulin and beta tubulin. So now we have to know how this dimer, it will be arranged in order to form the whole cylinder, right? This is just a unit, okay? This is 1 unit. I need more than that to form a microtubule. So how these, we can say, molecules, they will arrange. Yes, how they will arrange, how they will attach to each other to form the microtubules. All right. So it's very, very simple. Okay, between bracket, we can say it's simple. So imagine you have the dimer. Okay, you have alpha and beta. So what is gonna happen is that we're gonna have to alternate this dimer. So I have alpha beta, then I'm gonna have alpha beta, and then I'm gonna have alpha beta. So we're gonna have superposition of dimer. Okay? كل مرة I'm gonna add a dimer. This is a dimer, يجي another dimer, it will superpose to the previous one, and so on. Okay? Until they will form a filament which is called protofilament. Okay? So as you can see here in the pictures, I have my dimer. So this dimer, they will alternate. So this is dimer, second dimer, third, fourth, and so on. So all these, they will form a filament, which we call it the protofilament. To form a microtubule, the whole cylinder, I need 13 protofilament. I need 13 of this to form the whole cylinder. Okay? Is it clear? Or shall I repeat? Okay. So, I have dimer. Imagine I have 2 balls which are connected to each other, right? So these 2 balls, there is other 2 balls in the cytoplasm. They will come and they will superpose. Other 2 balls, they will come and they will superpose. So we're going to have like dimer, يجي فوق another dimer, then another dimer, then another dimer, until we're going to have like a filament. Okay? So this filament, it is called protofilament. Okay? So what you see here, this is 1 protofilament. Okay? To form the whole cylinder, the hollow cylinder, I need 13 like that. 13 protofilament like that to form the whole structure. Okay? Clear? Is it clear? Okay. So, as we said, the microtubules, they are short short-lived microtubules. Okay, they are kind of short-lived, okay, protein fiber. Yani they will be formed to ensure functions, and then, for example, they will leave. So these, we can say, microtubule, if we need them, if the cell need them, so they will be synthesized, right? Correct? So we're going to have growing formations of your microtubule to ensure, for example, the formation of the mitotic fiber, for example, okay? So once the mitosis is finished, so what will happen to the mitotic spindle? It will disassemble, okay? So the microtubule structures, it will disassemble, all right? So we have what we call a dynamic assembly, disassembly, assembly, disassembly, all right? Depending of the need of your cell, okay? If the cell need it, we're gonna assemble. If the cell then doesn't need it, we're gonna disassemble these structures. So we have like a dynamic of assembly and disassembly. And this dynamic, we call it instability, لأنه ما في something which is fixed, okay? It's a dynamic instability, it's unstable. It's not something once it is created, it will stay. This is, we can say, stable. Unstable, so one time is assembly, one time is disassembly. So it's not stable, okay? So it's and there is a dynamic, all right? This is dynamic of the cell, and it follow, we can say, the need of the cell, okay? Is it clear? Do you understand what is dynamic and stability? Yes.
It's just for the short-lived.
Yes, only for the short-lived. Okay? The ones which are there, they will stay there. All right? Yes? Dynamic, okay. Other questions? What is the other question? Yes?
The short-lived and the long-lived are also made out of protofilament tubule or different structure?
Yes, yes, all of them, they are made of tubulins. The long-lived, the short-lived, they are all made of the same thing. The thing between, for example, the short-lived, there is this dynamic instability of assembly, disassembly, assembly, disassembly, appear, disappear, we can say. Okay? So the dynamic instability, for the lady who didn't understand it, so we said that the protofilament or the microtubule, the cylinder, okay, or the mitotic, we can say, the spindle, for example, it appears, for example, for a period of time, okay, during mitosis, right? And then once the mitosis is finished, this mitotic spindle, it will disappear. So I'm going to have, during mitosis, I'm going to have assembly of tubulin. I'm going to form this microtubule. I'm going to form this mitotic fiber, okay? Once the mitosis is finished, is over, so what I will do, I'm going to disassemble all these structures. I'm going to disassemble all this, you can say, protofilament. I'm going to have free tubulin in the cytoplasm, okay? So this, we can say, alternations of assembly, disassembly, assembly, disassembly. It's what we call a dynamic, لأنه في حركية, we can say, a dynamic, but it is unstable because always we're going to change the status. One time assembly, one time disassembly. Okay? Clear? واضح? All right. All right. So now how this assembly will occur. Okay, so we said I'm going to alternating dimer over and over and over and over. You know, I'm going to superpose them one over the other. But how? Okay, is it random? No, it's not random. Okay, there is a mechanism for that. And you need to know this mechanism, how this assembly it will occur, how this superposition بتصير. Okay? So, as we said, the alpha and beta tubulin dimer, they have, we can say, We consider them a pole, right? So the beta tubulin, we call it the plus end. And the alpha tubulin, we call it the minus end, okay? And we have alpha and beta. This is plus, beta is plus. Well, alpha is minus. This is the plus end, the termination end, and I have the minus end. So the alpha is the minus, and the beta is the plus, okay? So how this assembly will happen? Imagine you have alpha, beta. I have beta on the top, alpha down. I have plus on the top and minus down. So the addition of the dimer, it's always from alpha to beta. So دايما the addition of the dimer بيصير over the beta. It will not happen from the alpha. So that's why plus and, as if I'm going to add plus, plus. So always the superpositions of the dimer بيصير on the beta side. Okay? Clear? So the growing from alpha, beta. So always we have alternation. For example, here alpha, it's in pink, beta in blue. So alpha is down, beta is on the top. So always I'm going to add alpha, beta, alpha, beta. Always beta is on the top. Okay? Clear? Clear so far? Before I move more, because it's getting, yes?
We will have 13 holes diameter or 13 per line.
I don't get your question. Can you repeat, please?
Will we have 13 holes or diameter per line, or we will have 13 lines assembled as?
Yes, we're going to have 13 protofilament, 13 complete, all right, to form the whole cylinder.
We don't know how much one line has of diameter, how many diameters of one line?
We cannot say how many because they are short-lived. They grow, and then after they shrink. They grow, and they shrink. Yani once they reach, for example, their target, so the synthesis, it will stop. Once they didn't reach, they will continue to assemble. Yani there is no number, exact number of the length or how many dimer we need to form a microtubule. Okay? So it depends what is the function of this microtubule, what we'll do inside the cell. Okay? Clear? Other question? Yes.
When you said that alpha and beta, we always add beta to alpha. That means that alpha is the basement of?
No, alpha هي البيزمنت. alpha من تحت and you have beta on the top. Okay? And دايما بنضيف imagine I have alpha beta, alpha beta, alpha beta. Okay? دايما the addition is from the beta. ما بيجي من تحت وبحط alpha beta. Okay? Clear? Yes.
And the plus and minus, does it refer to the charge?
No. It's just additions of stuff. Okay? Any other question before I move more in detail? Is it clear? Okay. So, the second details that I need to give you is that the alpha and beta subunits, the alpha and beta dimer tubulin, they have a molecule which is associated to them, which is called GTP. You know what is GTP? Guanine triphosphate. It's like ATP. It's another form of energy. Right, Kima? As maybe you know ATP. GTP is the same thing. Okay? So a GTP molecule, it is attached to this dimer. So as you can see here, for example, I have my dimer, for example, alpha and half beta, and I have this red dot, which is my GTP molecule. So each molecule of tubulin, not each dimer of tubulin has one GTP attached to it. Okay? Clear so far? because it's very important to for the formation, we can say, or for the assembly of the protofilament, each dimer needs to have GTP. If they don't have GTP, if this GTP it is hydrolyzed, this dimer it cannot be added. It cannot participate to the formations of your protofilament. So all the dimer, they must have GTP molecule associated to them. Okay? Clear? Yes.
Why ATP is not used instead of GTP?
This is GTP. This is the cell. So I did not create it. Yes.
GTP acts as a bond between alpha and beta.
It's not like a bond. It's like it's not a bond. It's not a bond. GTP is a source of energy. It's something that helps the dimer to, to associate to the protofilament. But GTP is a source of energy. It cannot be a bond.
Detached, it also loses one molecule, so it will be TD.
Yes. If it's detached, it will be converted. Yes.
What's the difference between ATP and ADP?
Okay. So GTP, and we have, for example, we have ATP, GTP, CTP. Okay, so you have these kind of triphosphate molecules. So they are all, we can say, energy molecules. All right. So for example, ATP, the body uses the ATP, مثلا the muscles. All right, they use the ATP, and how they use it? They use it, and it will be converted into ADP. Diphosphate. بيكون triphosphate بيصير diphosphate. Okay. If I can use the ADP راح يتحول into AMP, monophosphate. Okay. I'm gonna remove also one phosphate group. Okay. So 3 with D with M is related to the number of phosphate attached to the molecule. Okay, adenosine triphosphate, cytosine triphosphate, okay, guanosine triphosphate. Same thing for the GTP. GTP, if the body uses it, it will hydrolyze it to get the energy, and it will be converted into GDP. Okay, clear? واضح? Do you have other questions? Yes.
So when it's attached, does it lose the GTP? Or
Yes. When it will be attached to the molecule, it's the whole thing. Alpha, beta plus GTP. ما في GTP, there is no association at all. They will not participate to the formations of the protofilament. Okay? Yes.
So you're saying the dimer has GTP, but once it splits, the GTP becomes GDP?
شوي علي. Yes, I'm coming. Yes. خلاص? Okay. So now, as your colleague said, when the molecule, for example, as you can see here, we are adding, all right? I have here a dimer, then another dimer, then another dimer, then another dimer. And you can see here, the red dot here, it disappears. Okay? مو معناها ما عاد في GTP. The GTP was hydrolyzed and it is was converted into GDP. So once your dimer, they will be attached. So imagine I have here alpha and beta, and they have GTP. So they come and they want to participate to the formations of the protofilament. So they will associate, okay? And this GTP that they have, it will be hydrolyzed with here GDP. Okay? واضح? All right. So you will tell me, doctor, you said the molecule of tubulin, if they don't have GTP, you said, so they cannot participate. They will not be, involved in the structure, right? This is what I said, right? But what you have to keep in mind, that once the tubulin, they are involved in the structure, they are trapped. They are trapped in the structures, and they cannot move out. Okay? Is it clear? Okay? So to assemble, to form the microtubule, I need GTP. Once this dimer participate or it is added to the growing protofilament, this GTP, it will be hydrolyzed into GDP. But this dimer, it will stay in the structure. It will not move even though it has GTP now, okay? Because it is trapped in the structure, okay? So what's happened? So all these, they have GDP, all right? And this is the growing. Always you are adding. And this one, it is hydrolyzing, growing, hydrolyzing, and so on, okay? It's like an escalate, all right? So we are hydrolyze and addition, hydrolyze and addition, hydrolyze and addition. So it's growing, 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 growing of the microtubules. So what happened here at the end, we have what we call a GTP cap, as if it's a cap, all right, of your microtubule. This cap is a part of your, it's the top, we can say it, of your microtubule, and it contains only GTP, so that's why سميناه GTP cap, all right? So who needs a GDP? كلهم. But here we have the GTP cap, and this GTP cap, it is very important for the stabilizations of your microtubule. It's very important for your microtubule to not disassemble, okay? So to stay there, to maintain the structure, I need this GTP cap, okay? It's protection for the structures, okay? If this GTP cap, it will disappear. If all these GTP, it will be hydrolyzed and it will be converted into GDP, all the structures will disassemble. Okay? So now you will tell me, how can I know? Assembly, disassembly. How can I know that this microtubule, it's a growing microtubule, or if this microtubule, it will disappear? Depends. If the rate, the speed of the reactions of hydrolysis of a GTP, it is less than addition of a new dimer, I'm going to have a growing microtubule. بالعكس, if the hydrolysis بتكون أسرع, it will be more speed, okay? The rate of hydrolysis of GTP into GTP, it's more than the addition, I'm going to lose this GTP cap, I'm going to have a disassembly. Is it clear?
So the GTP cap has to always be more than the GDP.
يعني دايما speed of addition, السرعة addition of a new dimer بتكون أسرع من hydrolysis of the GTP. Okay? So the hydrolysis, if the hydrolysis is more speed than the addition, I'm going to have disassembly. If the kernel addition, it's more speed than the hydrolysis, I'm going to have growing. Is it clear? واضح? Yes.
Why is the first part become GDP, but the other side, although they are attached, stays as a PCB, not become a GDP?
Because it have to continue to grow. All right? This is a growing microtubule. Okay? If you need your microtubule to grow, it will be longer, we can say, we need this GTP cap. It will protect your microtubule from the disassembly. All right? If you remove this GTP cap, your microtubule will disassemble. Okay? Clear? Yes. Huh? The red molecules? Yes, the GTP. This is the GTP. الأحمر هذا, it is written here. Tubulin molecule bound to GTP. So الأحمر هو GTP. Okay? Have a question? Yes? يعني it's not really in the beta. If I go to the structure here, mostly it can be, okay? So, but you cannot say it's in the beta or the alpha because it's a dimer. They are together, all right? So as you can see here, I have alpha. This is alpha dimer, alpha monomer, and I have the beta monomer. Here it is attached to the GTP bound, okay, the GTP. And here after, once the GTP it is hydrolyzed, it will be here GDP, all right? So you can see here in the pictures. So all these, we can say, parts of the microtubule contain GDP. And this is the GTP cap, okay? It contains your dimer with the GTP, okay? Yes?
What should happen when the microtubule becomes disassembled?
I'm coming. Yes?
The alpha and the beta are stick together. Like, are they stick or adhered as like one molecule? Or
Yes, yes.
separate?
No. الاثنين مع بعض. بالاثنين 2 2 2. Okay?
So they become one.
As if it's one structure. It's one structure. This is the unit, the structural unit. Okay? So it's one unit. Okay? Other questions before I move? All right. Is it clear? واضح? Okay. So I'm going to show you now a short video, so which will show you, we can say, the assembly, all right, of the tubulin dimer, okay?
Microtubules are one of three principal components of the cytoskeleton. A microtubule is composed of tubulin dimers, consisting of two closely related polypeptides called alpha and beta tubulin. Tubulin dimers polymerize into protofilaments assembled around a hollow core. Tubulin dimers polymerize at the plus end of a microtubule. The polymerized tubulin dimers form a sheet that zips up into the mature microtubule just behind the region of growth. The tubulin dimers that add to the microtubule are bound to GTP. The tip of the microtubule contains tubulin dimers that are also bound to GTP. The rest of the microtubule consists of tubulin bound to GDP. Shortly after new tubulin dimers are added to the microtubule, GTP is hydrolyzed to GDP. The hydrolysis of GTP follows the growth of the microtubule. If the concentration of GTP-bound tubulin decreases, new subunits are added more slowly, and GTP hydrolysis catches up to the end of the microtubule. GDP-bound tubulin has a weaker binding affinity for its neighboring molecules than GTP-bound tubulin does, and the presence of GDP-bound tubulin at the end of the microtubule leads to its disassembly and shrinkage. The alternation between cycles of growth and shrinkage is called dynamic instability.
Okay? Is it clear? So I just show you how the protofilament, they are assembly, how the speed, for example, the ADP catch, the hydrolysis of GTP catch the synthesis, how the protofilament, they will disassemble, because these molecules, when they have GDP, as we said, they cannot bind to each other. They don't have any, he said, affinity, all right, to each other, right? So that's why they will disassemble. So here in this slide, we just show you again the disassembly process. Disassembly process, as we said, when your molecules of microtubule, it will lose its, we can say, GTP cup, all right? When the hydrolysis of GTP, it is more speed, all right, than the additions of new molecules of tubulin. All right. So that's why if it will reach, we can say, to the GTP, we don't have any more GTP cap. What will happen? This protofilament, they will peel back. They will curve away, as you can see here, the electronic microscopes, a microfilament, which is during its disassembly, all right? So they will peel back, and they're going to dissociate. We don't have any more microtubule structures, okay? Is it clear? All right. So here is just to show you the speed, all right? So if the synthesis of the new, we can say, protofilament, it is, we can say, faster than the hydrolysis. I'm going to have, a growing microtubule. However, if the hydrolysis of GTP into GTP is faster than the addition of new tubulin, so I'm going to have disassembly. Okay? All right? So it's a balance between these two reactions. All right? Either I have assembly or disassembly, depend of the speed of these reactions. Okay?
If they disassemble, this means that their function is done.
Yes. Finish.
And this thing is microtubule, right? And does it always have to have a GTP cup?
As growing, yes. As growing, yes. But once it's finished, it reached, we don't have any more growing, all right? The structure is there. So after we're going to reach, its functions, it will disassemble, okay? Long-lived, they are assembled, خلاص, they are there. With the the GDP, not GTP, yes. Yeah.
Will the molecules disappear then, or will the process start again?
Of course. And it will disappear, but it will not disappear as filament. All right? It will be completely dissociated. So we're going to find in our cytoplasm tubulin, free tubulin, not free protofilament. All right? The protofilament, they will assemble when you're going to have, when you need them. So the disassembly, it's completely at the unit structure level. All right? So I'm going to have tubulin released in the cytoplasm. Once we need, again, this, we can say, microtubule. So this tubulin, they will assemble, they will form protofilament, and they will form microtubule. Okay? واضح? Okay. So, all right. So we finish by this, we can say, slide. Remember when we spoke about microtubule function, we said that the microtubule, they help in the cell motility. Okay? So, these microtubule, as you can see, this is a microtubule. So I have here a microtubule, all right? And this microtubule, it will be used by specific protein, which we call them motor protein, all right? So in our cytoplasm, we have a motor protein. The protein هذول, what are their function? Function is the transport of vesicle, is the transport of organelle from one side to another side of the cell. Okay, we said that the vesicle, the organelle, they are stuck, they are anchored inside the cytoskeleton, correct? Am I correct? Sure. You have to be sure of yourself. I said that the organelle.
Muscular.
Muscular? What do you mean muscular? I said that the organelle, they are attached, they are anchored to the cytoskeleton, right? So they have a fixed position. Okay? Correct. So what if one vesicle wants to move from one side to another side?
By motor protein?
Yes. I need motor protein. Motor protein, they are carrier, okay? They are the transporter, all right? They carry the vesicle or whatever from one side of the cell to another cell. But they will use the microtubule as their route. طريق تبعهم, okay? Is their truck, all right? They will use this, and of course, here you have, for example, we have two types. We have kinesin, it's one motor protein, and you have the dynein, it's another motor protein. And the kinesin, they transport the molecule to the plus end of the microtubule. However, the dynein, they will transport to the minus end of the microtubules, okay? So this is very important. And of course, if they are, we said, motor protein, so they need energy, okay, to move. And this energy, it is given by ATP. So always they have in their head, we can say a site for ATP binding, okay? We have ATP in their site. I'm going to show you a video where we're going to show the kinesin here, protein, working on the microtubule. I hope I can work it. Open. Here. So this is your kinesin, and this is the big organelle, is the vesicle, and this is your microtubule. So you see how it's carry, you know, your vesicle along your microtubule. This is the network of the cytoskeleton. You have the organelle and so on. It's clear? خلاص. Thank you very much.
انا كنت بدرس لكم وكان في شيء ما فهمته بالسلايدات. شنو الفرق بين النيوكليوس اللي هي النواه